Every protein in your brain is wearing a tiny sugar tag, a label telling your cells where to send it and whether to trust it. Scientists just found a supplement millions take for their joints jams that labeling system into overdrive. Here's the thing about your brain. It doesn't just build proteins, it labels them. Almost every protein your neurons make gets a cluster of sugar molecules stuck onto its surface. Scientists call this glycosylation. And that sugar label works like a shipping tag, telling the cell where the protein should go and which other proteins it's allowed to interact with. Get the labeling right, and your neurons fire, remember, and repair themselves the way they're supposed to. Get it wrong, and the whole delivery system backs up. Alzheimer's affects tens of millions of people worldwide, and for 3 decades, most Alzheimer's research has chased one suspect: sticky amyloid plaques. A brand new study just found the brain's labeling department might be just as guilty, and it points a finger at one of the best-selling supplements on Earth. That supplement is glucosamine, usually sold with chondroitin in a bottle promising stronger, more flexible joints. Genuinely one of the biggest sellers in the entire vitamin aisle, with worldwide spending projected to climb toward $3.5 billion a year by 2025, according to Harvard Health Publishing. You'd assume the science backs that up. According to rheumatologist Robert Schmerling of Harvard Medical School, it really doesn't. A 2018 review found only small, questionable improvements in pain. A 2022 analysis, pooling 8 separate studies and nearly 4,000 people with knee osteoarthritis, found no convincing evidence the supplement does much of anything. And Schmerling notes the case for it protecting joints from wearing down in the first place is just as weak. Here's the detail that should have made bigger headlines. One landmark trial, giving glucosamine and chondroitin to 164 people with knee pain, got stopped early. Not because it worked too well, but because the group taking the supplement reported worse symptoms than the group on a sugar pill. The bottle promises joint support. The evidence is shaky at best. So if glucosamine isn't doing much for your knees, what is it doing once it's absorbed into the rest of your body, including your brain? That's exactly what a team at the University of Florida, led by researcher Ramon Sun, set out to answer, publishing their results this year in the journal Nature Metabolism. They scanned real human brain tissue from Alzheimer's patients using imaging technology that maps thousands of molecules across a slice of tissue at once. In the Alzheimer's brains, one signal stood out: an explosion of sugar tags plastered across proteins in both gray and white matter. Not a little more, dramatically more. The team graded each brain sample using Brock staging, a scale doctors use to measure how far Alzheimer's has spread, running from 0, no disease at all, up to 5 and 6, the most advanced stages. The sugar tag overload climbed in lockstep with that scale. Healthier brains carried fewer tags. The most ravaged brains carried the most, stacked on thick. The team ran the same scan on 2 strains of Alzheimer's mice, one bred to build amyloid plaques, the other bred to build tangled tau fibers, the 2 proteins most often blamed for the disease. Both mouse types showed the same tag pileup, different broken protein, same overworked labeling machine. To figure out why the tags were piling up, the team ran a tracing experiment, feeding mice a trackable, heavier form of carbon-labeled glucose and watching where it ended up. If the brain were simply failing to clear old tags away, the labeled sugar wouldn't show up faster than normal. Instead, Alzheimer's mice packed far more tracked carbon into freshly made sugar tags than healthy mice did, while old tag clearance barely changed. This isn't a cleanup problem, it's a production problem. The labeling department is running the printing press harder than it should, even as the rest of the brain is starved for the glucose that PRESS depends on. So Sun's team took it a step further. Is the labeling overload actually driving the damage, or just riding alongside it? They went after the machine printing the tags. One tool, a genetic dial-down of an enzyme called PGM3, essentially the printing press for these sugar labels, cut tag levels in the brains of Alzheimer's mice. A second tool, a drug that physically jams the machinery attaching tags to proteins, produced the same drop through a completely different route. Both interventions, tested independently, led to the same result. Memory improved. Amyloid plaques and tau tangles barely budged. The benefit was coming from somewhere else entirely, from quieting the label printing itself, not from clearing the usual suspects. Why would a starving brain choose to spend its limited glucose printing more labels in the first place? The researchers' working theory: it starts as a rescue attempt, stressed neurons slapping extra tags onto proteins to keep them stable while energy runs low. Useful briefly. Left running for years, it becomes its own problem. Then they ran it in reverse. Instead of cutting the tag supply, they fed Alzheimer's mice oral glucosamine at a dose calculated to match the roughly 2,500 milligrams a day a person takes for their joints. Glucosamine is, chemically, raw material for building these exact sugar tags. In the Alzheimer's mice, flooding the system with extra raw material pushed the labeling machine further into overdrive, and their memory got dramatically worse. Worse than untreated Alzheimer's mice. Here's the part that matters just as much. The identical dose given to healthy mice did nothing. No extra tags, no memory change. A healthy brain apparently has some built-in regulator keeping the labeling system in check. An Alzheimer's brain has already lost it. Mice are one thing. So Sun's team pulled real medical records. More than 50,000 patients diagnosed with Alzheimer's dementia and another 41,000 with mild cognitive impairment, the fuzzy in-between stage before a full dementia diagnosis. About 8% of each group had documented at least a year of glucosamine use. Among patients with full-blown Alzheimer's dementia, glucosamine users had a 25% higher risk of dying over the next 10 years. Odds of that being random chance under 1 in 400. Here's the split almost nobody would expect. That mortality difference didn't show up in the mild cognitive impairment group at all. What showed up there instead was a 25% higher rate of progressing from mild impairment into full Alzheimer's. Two different populations, two different consequences. This is observational data. It can't prove glucosamine caused those numbers by itself, but it lines up, almost too neatly, with what the mice already showed. The harmful signal in this study is specific. It only showed up in brains already deep into Alzheimer's or sliding toward it. Healthy mice given the identical glucosamine dose shrugged off the extra tags completely, memory unaffected. What the study hands the field instead is a testable drug target sitting outside the amyloid research that's produced 30 years of disappointing trials. Turning down that one overworked labeling machine improved memory in mice without touching a single plaque or tangle. On a disease that's been mostly closed doors, that's a new one standing open. If this made you rethink your medicine cabinet, subscribe.